Welcome to my presentation on how hippocampal neurons code individual episodic memories in humans. Let's jump right in. We know that the hippocampus is essential for episodic memories, but there are two possible memory codes. Now, one has been proposed 40 years ago by Tyler and Senna, and it basically states that an episode, for example, here, you're sitting in your favorite cafe with your best friend, and a waiter is attending another table and there's music in the background. So multiple elements. And according to Tyler and Senna, all of these elements are bound to one index assembly, one assembly of neurons in the hippocampus. Now, as an alternative, Quiroga has proposed in 2012 that each of these elements are coded by one individual concept neuron. And in this case, one episodic memory would be coded by the concurrent activity by multiple of these concept neurons. Now, we investigated this conundrum using Benker-Fried electrodes implanted in epilepsy patients. On the left, you can see one of those Benker-Fried electrodes, and at the tip protrude 9 microwires. 8 are used for recording and 1 as a reference. Here on the right, you can see at the top another schematic of the same electrode, and at the bottom you can see how it looks like in a scan once they're implanted. Now, mind you that they look much bigger in the scan, but this is because they're made out of metal and produce a large artifact. What we then get from this recording is first raw data, which is just the LFP you can see here, so lower frequency, and on top of this are transient high amplitude reflections. So those are the spikes that we're looking for. We can then filter the raw signal to get the spike band, which is usually around 300 to 3000 hertz, and apply a simple thresholding approach to then identify these spikes. Because we sample this signal with a very high sampling rate of about 32,000 hertz, we can then basically zoom in into each of these spikes and use this spike wave shape to then cluster each spike into a putative single neuron. You can see an example of 2 different single units here on the right. Now, unfortunately, we can't send our patients to the cafe when we want to investigate their episodic memory. So instead, we have a very simple memory association paradigm. Here, we present the patient with an animal cue, in this case, a tiger, and then 2 seconds later, 2 associate images come on screen. Now, these could be either 2 faces, 2 places, or a face in a place. This is going to be important later on. Every single event is encoded and retrieved only once, and it's self-paced. So the patients have as much time to come up with a vivid story, including all 3 images as they want to. Now, during retrieval, we present them again with the animal cue, and the patients have to detect out of an area of 4 images the correct 2 in order for it to be a correctly remembered trial. We have a rather large data set of 16 patients, 59 sessions, and a total of 585 neurons, all located in the hippocampus. You can see here an analysis schematic. On the y-axis, you can see the standardized firing rate, and on the x-axis, you can see the trial number. Now, color-coded in purple is encoding, and retrieval is in orange. This is for one particular single unit. And we define the reinstatement as the product of the standardized firing rate during encoding times retrieval in one particular trial. So you can see right away there's a lot of firing reinstatement only in trial 4 here. In order to threshold this and look where we have a relevant reinstatement, we then use the shuffling procedure and computed the 99th percentile. So this could look something like this. There's a threshold at 4. And once there's a significant or above threshold, rather, firing reinstatement, we would consider this an episode-specific neuron because it's reinstating its firing rate that it had during encoding. And this is an example episode-specific neuron, so you have a rough idea how it looks like. This is just a raster plot of the firing of this neuron. On the y-axis, you have the trial number, and on the x-axis, you have the time. Color-coded again, purple encoding and orange retrieval. And I've adjusted also the transparency according to the reinstatement values. This is just a visualization feature to make it easier to focus on the relevant trials. You can see here trial maybe 7 and 22, 23 here seem to have a lot of reinstatements. Now, if we only look at the reinstated trials, this is the instantaneous firing rate, again color-coded in purple for encoding and orange for retrieval. Here on the right, you can see the reinstatement values, so high reinstatement values for those two trials, and you can also see the animal cues and the two associated images here and here. Another interesting thing is we have quite a nice wave shape for this ESN. And importantly, it's only firing about 1,100 times in a recording that typically takes 40 to 45 minutes. So it's firing very sparsely. Now we have 585 of these neurons. So just by chance, we might have a lot of these episode-specific neurons. So what we did is we would apply a second-order permutation test to see how many episode-specific neurons we would expect under the null. And this is what you see here on the right. So this is the distribution of ESNs under the null. And here on the right is how many episode-specific neurons we found empirically. So in total, we have 141, roughly a fourth of all recorded neurons that are identified as episode-specific neurons. But now, all of these episode-specific neurons might be concept neurons that are tuned to the associate images. It might be that these neurons are literally Jennifer Aniston neurons. We have to somehow rule that out. And we do this by first leveraging the experimental design. If we have episode-specific neurons that are coding to one trial with two places and also another trial with two faces, we can rule out that it is a concept neuron because in one trial it's two faces and another two places, it's a non-overlapping stimulus material. And when we do this, it's a very conservative approach, of course, because not many episode-specific neurons fire in response to multiple trials. But we still find a significant number of these episode-specific neurons under this new criterion. So our results cannot be explained by just concept neurons being tuned to the associate image. Furthermore, episode-specific neurons are limited to later remembered episodes. So what we did is compare how many episode-specific neurons we find if we limit ourselves to later forgotten trials and contrast that with the number of episode-specific neurons we find during the later remembered trials. And we find that if we limit ourselves to later forgotten trials, we have a significant lower number of episode-specific neurons. This means that we cannot construe our episode-specific neurons as concept neurons tuned to the associate, because the associate is only in the head of our participants during retrieval. Now, you might play devil's advocate and say, what about concept neurons that are tuned to the animal cue? And I would have to agree with you. What if we have episode-specific neurons that are in this case tuned to the tiger, would have the same results potentially? So because of this, we have acquired a new data set, which is largely the same in design when it comes to the memory association tasks, but we add a visual tuning task at the end. In this visual tuning task, we take all of the stimuli that we have previously used in the memory task, and we show it repeatedly without any memory task. This is an approach that has been used traditionally to identify concept neurons. So in this data set, we have a total of 8 patients, 13 sessions, and 86 neurons all located in the hippocampus. And we could replicate our results. So now again, we could identify in this second experiment concept neurons and then discard them from our analysis. But if we do this, we can rule out concept neurons tuned to the animal and also to the associate image. And we can still find a significant number of episode-specific neurons. It's a little bit less than a fourth in this case. Another thing you might say is maybe episode-specific neurons are time cells. So time cells would be single neurons that code for specific reoccurring time points. However, we did not find a significant number of time cells in our data set. And critically, there was no significant overlap between the time cells that we did identify and our episode-specific neurons. Now, mind you, our experimental design was not meant to uncover and identify time cells. However, these results do mean that our episode-specific neurons cannot be construed as time cells. Another interesting thing as well is that these episode-specific neurons, on average, have a wider wave shape. And the wider wave shape has been previously shown to be associated with excitatory cells. And you can see here down below a distribution of the spike width on the x-axis and the probability on the y-axis, and in blue, light blue, episode-specific neurons, and in a light green, the spike width of single neurons. So to conclude, we have single neurons in the hippocampus that exhibit a memory-specific reinstatement of neural firing for individual episodes. Now, this contains two important pieces of information. First, we have single neurons and not a population code of all recorded single neurons. Here we have individual single neurons. And the other thing is they code for individual episodes. They don't code for successful memory as a whole, so for very specific trials only. And these episode-specific neurons cannot be explained by means of equating them with concept neurons or time cells. And on top of this, they seem to be excitatory neurons. So thank you for your attention. And you can find the current version of the manuscript on bioRxiv. And you can find me on Twitter at Luca Kolibius. If you have any question, please feel free to shoot them my way. Thank you.